Hello, I'm Professor Paul Bingham. This is Biochemistry 1, and our goal in this segment is to take the first steps toward mastery of a uniquely interesting enzyme called pyruvate dehydrogenase, PDH for short. This enzyme is not only especially central to the flow of metabolic carbon through the processes that drive anabolism and catabolism that drive our continued existence, but it has some quite unique or quite unusual properties that will teach us some really valuable general lessons about coenzymes that participate in energy metabolism, coenzymes that we're going to encounter again and again after today. So remember our broader context. Organisms, we can think of them as vehicles, whether they're oak trees or human beings or bacterial cells. They're built by design information, and they behave as if the replication of that design information were their purpose, and they build all kinds of tools to make that possible. And the tools that we've been focusing on for quite some time now in this segment are the molecular machines that represent catalysts, that catalyze chemical reactions. And among a subset of those molecular machines, those catalyze a specific set of reactions, which is the high-speed flux of massive amounts of carbon and, more importantly, energy through living organisms, fueling our movements and our thinking and all the things that make us alive, and at the same time providing some carbon for building block molecules to make the spare parts, the replacement parts that we need as molecules wear out in our daily adult life. The collective flow of all that matter, catalyzed by tools, by enzymes, including PDH, through organisms collectively, as we've said several times before, is referred to as metabolism. The metabolism that we're mostly going to be concerned with today is still part of catabolism, that is the burning of high energy food that we take in, oxidizing it ultimately to CO2 and water to liberate the energy that it contains, the energy that will then be stored in various currency molecules for then subsequent use in anabolism and other things that we'll come to in later segments. Again, this slide you've seen before just emphasizes that hydrocarbons generally have reduced hydrocarbons like gasoline or the food that we eat have lots of energy, and as we oxidize them by combining their components with molecular oxygen, we release a lot of that energy, oxidizing them ultimately to CO2 and water. And as you'll see, in fact, one of the things that PDH does is liberate one of the CO2 molecules from the original parental glucose that will ultimately be reduced all the way to CO2 by the time we get done in the next few segments with its metabolism. So let's put PDH in context. I alluded to its uniquely central role. So this is a typical animal cell with a large cytosolic component, the sort of aqueous environment in which all the other cell components are embedded, and the mitochondrion, which is in fact where PDH lives and acts, and where a number of things that we'll talk about in the next several segments also occur. The mitochondrion is a complex two-layered, surrounded by two membrane layers with a complex folding pattern illustrated here. We'll have much more to say about that later. All that you need to be aware of right now is that PDH is in the inside of that second inner part of the mitochondrion, referred to as the mitochondrial matrix. That's where PDH lives. So let's now put PDH in context. So we've talked in the preceding segments about glycolysis, how glucose is broken down to pyruvate with the extraction of energy, as we've again talked about previously. That pyruvate is then going to move into the mitochondrial matrix, and the first thing it's going to encounter is PDH, the subject of our discussion today. So in other words, PDH sits right at the interface between cytosolic glycolysis and the beginning of the mitochondrial parts of metabolism that we'll talk about later, and I'll remind you briefly about in the next few minutes. So this is a chart just summarizes in a very simplified form the flow of glucose and some other molecules I'll call your attention to in a moment, all the way through to carbon dioxide, and not shown here, water. In other words, the complete oxidation of the reduced hydrocarbon glucose to CO2 and water. As we saw before, glycolysis goes on in the cytosol. Everything after glycolysis, including the fate of pyruvate, goes on in the mitochondrion. And it all feeds into the citric acid cycle, or sometimes called the TCA or Krebs cycle, illustrated by the shaded circle at the bottom. Before we dive in to the metabolism of pyruvate by PDH, which is going to be our subject today, I want to put PDH in context in another way. So it's the transition from cytosolic glycolysis to the TCA cycle. But notice what its product is, the TCA cycle shown here. Its product is, one of its products is CO2. So it's releasing a CO2 molecule, something we talked about a moment ago. But its other major product is acetyl-CoA. And I want to call your attention to the significance of acetyl-CoA for other subjects we'll talk about later before we return to its significance in the context of Pdh function. So carbohydrates, as we've already said, are typically reduced to glucose, and then the glucose circulating in the blood circulatory system taken up by your cells is burned through glycolysis to pyruvate, and that will then enter the Pdh complex in the TCA cycle, as we'll see in a moment. But notice here that the fatty acids generated from the catabolism, the breakdown of lipids, end up producing acetyl-CoA, as do a number of amino acid products feed in either as acetyl-CoA or into other parts of the TCA cycle. So this central simple metabolic diagram captures a great deal of what's going on with regard to catabolic metabolism in animals. Again, our focus here is to understand the role of glycolytic pyruvate passing through PDH in preparation to enter the TCA or Krebs cycle. So Pdh catalyzes that reaction, as we've just said. So here is the Krebs cycle itself. This looks a little intimidatingly complicated. Don't worry, in later segments we'll go through it in detail. It's actually 8 simple steps that you can, in fact, understand quite well and have some really interesting lessons to teach us. But our concern today is just the Pdh reaction, which is going to grab pyruvate and turn it into acetyl-CoA to let it enter the citric acid or TCA cycle. So let's blow that up, and let's look at the sort of gang of reactants and products that are going to participate here. So we have obviously pyruvate itself. Pyruvate is processed by Pdh to release a CO2, as we've already said, circled in red. And the other 2 carbons end up forming acetyl-CoA, about which we'll have a great deal more to say in a few minutes, circled in red here. The coenzyme A is a coenzyme, is a cofactor that cyclically participates in a kind of covalent catalysis, as we'll see later. And then lastly, oxidized NAD, it's actually NAD plus, has a positive charge on it, is reduced to the neutral NADH compound with the capture of reducing potential, something we saw transiently in the context of glycolysis, but that's going to be much more central to energy metabolism moving forward from here. We're not going to talk too much today about what happens to the reducing potential from Pdh. We'll talk about that a little bit later in later segments, but it's going to turn out to be really a central issue with regard to how energy metabolism works. That is where reduced NAD goes. All right. So let's now zero in a little more carefully on the Pdh catalytic cycle. Before I do that, let me call your attention to the fact that acetyl-CoA in the subsequent reaction, the citrate synthase reaction diagrammed here, are joined, spending the energy embedded in that high energy bond between acetate and CoA, as we'll see a little bit later, to make citrate, to start the citric acid cycle, as it's called. But again, our focus today will be not that, but rather how you get the acetyl-CoA to start the process, Pdh. All right. So let's look at an overview of the reactions. First, let's just look at the basic chemistry. So you start with pyruvate, coenzyme A, and an oxidized NAD, and you end up with acetyl-CoA, carbon dioxide, and reduced NAD. It looks relatively simple at first glance, although you'll notice that there are 3 reactants and 3 products. In fact, the sequence of reactions that carries out this process are perhaps a little bit surprisingly complex, involving 3 separate enzymes joined together in Pdh. In fact, we often don't call it Pdh, we often call it the Pdh complex, for reasons that will unfold over the next couple of minutes. There are copies, multiple copies, of each of 3 different enzymes organized to execute this catalytic process. So here is a diagram of that. This diagram is a little intimidating at first glance, but be patient. Everything you need to know is summarized here, and we'll gradually work through and fill in each of the pieces. You'll see each of the pieces are straightforward and easy to understand, and each has lessons to teach us, as I alluded to at the beginning, not just about Pdh, but about energy metabolism more generally going forward from here. So first, notice, as I said, there are 3 enzymes. They're often, for economy, just called E1, E2, and you'll notice here that E2 is cycled twice, circled or boxed twice, once on the catalytic circle around the outside, but notice in the middle there's something called E2, E3BP. Well, in mammalian cells, there are a lot of copies of E2, and there are a few copies of an E2-like protein called E3BP, the E3 binding protein. We need not be concerned about that here. We'll just simplify this by pretending that the entire complex is built around multiple copies of E2 itself for simplicity. The crucial point here is that E2 has a dual role. It's the central